To design a drug that actually works, structural biologists have one ultimate target: locating a hidden, transient cavity deep inside a complex protein. We rely heavily on experimental data to map these structures. High-resolution cryo-EM provides incredible detail, but it only produces a frozen, static snapshot of biological reality. Inside a living cell, biomolecules are subjected to intense thermodynamic forces. They are constantly shifting, folding, and moving through thousands of different conformational states every second. If we want to understand how a disease mechanism actually operates, we have to look past the static model. We need to bridge AI-driven structure prediction with molecular dynamics to watch the protein move. That is the specific problem UCSF ChimeraX solves. It has evolved from a standard visualization tool into a complete computational ecosystem built to process both structure and motion in one place. When computational tools are fragmented, data gets trapped in silos. By bringing everything into a unified environment, we can decode the transient, fleeting states where disease mechanisms actually occur. Let's start at the end of the modern R&D pipeline. The primary objective is to find an actionable binding site, a pocket that stays open just long enough for a therapeutic molecule to attach. ChimeraX uses an enhanced cavity detection algorithm, deploying precise vector probes to scan topography and calculate subsurface void volumes. Simultaneously, the algorithm scores each void, mapping a numerical score directly to its localized electrostatic properties. This precise quantitative mapping allows computational chemists to immediately filter out non-viable targets, accelerating the lead identification phase of drug discovery. But there is a major complication. A protein and its baseline resting state almost never displays these optimally scored cavities. Because proteins are in constant motion, a pocket that appears closed in a static map might only exist for a few nanoseconds. To target it, researchers must identify exactly when this window opens during the protein's conformational shifts. This creates a massive data bottleneck. We are looking for a cavity that might only exist for a fraction of a microsecond, hidden within an enormous ensemble of constantly shifting protein conformations. To generate that thermodynamic data, ChimeraX now includes native support for the Bolt server, providing the computational engine needed to run comprehensive molecular dynamics simulations. In this 2D scatterplot, thousands of structural conformations overlap into raw data chaos. ChimeraX's clustering algorithm separates this mass into distinct color-coded islands based on structural similarity. We then isolate one sparsely populated cluster, representing the exact moment our target cavity opens. By isolating this specific trajectory data, we can take those mathematical coordinates and translate them back into a targeted, highly detailed 3D rendering of the active protein. Trajectory clustering serves as the critical bridge. It takes terabytes of raw thermodynamic output and distills it into a singular, actionable structural state. Running these molecular dynamic simulations requires a starting point. If the initial baseline structure is flawed, the resulting thermodynamic data is useless. In the past, deriving a single baseline structure meant relying entirely on slow, laborious experimental methods that could stall a project for months. ChimeraX solves this at the origin point through the native integration of OpenFold 3, which generates accurate structural baselines on demand. This diagram shows the speed of that input-to-output process. We start with a linear, 1D string of amino acids, which the AI rapidly predicts and snaps into a complex 3D backbone of alpha helices and beta sheets. This AI-assisted workflow gives researchers highly accurate protein folding predictions without ever forcing them to leave the software environment. Because it happens natively, the OpenFold 3 prediction feeds directly into Bolt's molecular dynamic simulations and ultimately cavity detection algorithms. Integrating AI structure prediction natively eliminates the friction between raw sequence data and structural simulation, creating a continuous, unbroken computational chain. We've mapped the pipeline in reverse. It begins with a raw sequence fed into OpenFold 3, moves to a Bolt's MD simulation, gets organized by trajectory clustering, and ends with precise cavity detection. ChimeraX unifies prediction, simulation, and analysis into a single environment, removing the bottlenecks of fragmented toolchains. As shown here, data pulses move seamlessly between prediction, simulation, clustering, and detection. This unified ecosystem replaces disjointed terminal scripts and manual file conversions with automated, real-time visual analysis. For pharmaceutical R&D engineers and computational chemists, this means identifying promising drug candidates faster, radically compressing the timeline from initial sequence to lead identification. By integrating AI-driven protein predictions directly with advanced thermodynamic visualization, UCSF ChimeraX provides the technical infrastructure required to increase the speed and efficacy of modern drug discovery.